Okay, so this is the notes that we're going over. The first part of this slide was the endosymbiotic theory. This was the article that you guys read. Basically, what it's talking about is that chloroplast, what it's talking about is that the chloroplast and the mitochondria were their own parts of the cell, and they lived outside of the cell. And what happened was this thing called endocytosis. And you see that happening over here in this first picture. The cell membrane is going around either the, what is called here is the bacterium, it's going around that, and it's engulfing it into the cell. And what happens then is that a membrane is formed around the cell as well. That's where we get our double membrane layer. And then after what's happening here as it goes through this evolution is this mitochondria came from this bacteria and it created its own inner and outer membrane. These ones are pretty easy. Robert Hooke first coined the term cells, so these are all these old dead guys that we're going to have to know. So Robert Hooke is the one that just coined the term cells. Brown is the guy that was first termed to see the nucleus, and he said that certain cells looked more dense than the others, and that's why they discovered that inside those cells was something. That something actually became the nucleus. Schleiden is the botanist, so he said that all plants are made of cells. Schwann is a zoologist. He saw that all animals were made of cells. Leeuwenhoek actually used a microscope to see that there were microorganisms or protozoans, and those are all unicellular organisms, and that they could live as just one cell on their own. And Virchow observed that these cells were dividing so that all cells came from pre-existing cells. They didn't just sprout up automatically. And all of these guys combined actually gave us the cell theory. All things have cells. Cells are the basic unit of structure and function in living things, and all cells come from pre-existing cells. All of those things make up our cell theory. We have two types of things, prokaryotes versus eukaryotes. Over here are all the prokaryotic functions. They are simple. They are very, very small. They have no membrane. They have no nucleus, and it's usually something like bacteria. The eukaryotes then are more complex. They're a little bit larger. They do have a membrane and a nucleus contained in them, and these are going to be everything else, the plant, animal, and the fungi cells. Prokaryotic cell structure, they are unicellular, but they can associate together in what's called like a cluster, so they can just be around each other and exist as this this area of being. There's just some bacterial shapes that we tend to classify them as, and then you can see this colony of cells that's over here. Prokaryotic cells, I gave you this slide, but this one is a little bit easier to talk about. Prokaryotic cells, it's going to have some sort of membrane, usually also a cell wall. Cell wall is going to be more structured and supportive. It also has this flagellum that's going to be something that's going to be used for propulsion to move around. Cytoplasm, of course, is contained within the cell wall. It's the jelly-like stuff that is suspending everything. Ribosomes, because they do have to make some proteins, because they also do have chromosomes and DNA. The plasma membrane is like the cell membrane. And then they also have these things called plasmids that are inside. Plasmids are not organelles. They're just something that exists inside of the prokaryotic type cells. Cell traits, most are bacteria. They can be beneficial, even though we think of bacteria as sometimes being bad. All ecosystems need these bacterial things, such as decomposers, to break down and recycle the nutrients. Prokaryotes are also called autotrophs because they make them their own food. They are primary producers. And some bacteria are helpful as sometimes antibiotics. Eukaryotic cells then are a little bit different. They were more complex. They have the organelles that are functional parts. So each organelle has its own specific function to help that cell exist. And each organelle also has its own membrane. So these are membrane-bound organelles on the inside of the eukaryotic cell. Compartmentalization is that nucleus is going to be enclosed in DNA, or the nucleus is going to protect the DNA so it doesn't break down when it has there. Organelles may make a substance that will later be consumed by another organelle. And then this also allows us to have the division of labor with different things doing specific functions. So if we start out with our cell membrane, or sometimes it's called the plasma membrane, it's the boundary, it's the gatekeeper, it has specific rules on what can and cannot go in. We also have things that exist inside the membrane of porous channels and proteins. We're going to focus on this a little bit more after 7.2, but these things are also going to be allowed to let things come in. Like over here I have written down ions use the protein channel in order to enter and exit because they're a little bit bigger. They can't diffuse straight through. We said earlier in the homework that there is a phospholipid bilayer. Phospholipid is the specific type of lipid that makes up the cell membrane, and it is a bilayer because it exists of two layers of lipids. They have something that's called a hydrophilic head and a hydrophobic tails. The hydrophilic end is polar, so it likes water, and the hydrophobic end is nonpolar, so it does not like water. It's set up in this configuration where the heads and on the outside and the tails are on the inside. The nucleus is going to be the boss or the organizer, the control center. It is also enclosed by its own membrane, the nuclear membrane. This is the genetic control center. It keeps and houses all of the DNA inside of the nucleus. That does have pores on the outside that allow RNA to pass through, because the RNA, we said, is the only spot that gets to go outside of the nucleus and take the directions to the ribosomes where it can make the proteins. The nucleolus is inside of the nucleus. It's another part of control, exactly where the DNA is housed. And then this is also something that's going to partially help to assemble the ribosomes. This is what you're going to look at to be able to distinguish between eukaryotes and prokaryotes. If it has a nucleus, it's a eukaryote. If it doesn't, it's a prokaryote. Cytoskeleton is made up of two types of fibers, microtubules and microfilaments. These are going to be the scaffolding, the shape. It's going to help hold some organelles in place along with the cytoplasm. And we're going to see a little bit later on that these microtubules are also going to be connected to the centrioles that allow the things to travel for cell replication. Lysosomes are the janitor of the cell. It cleans up the waste. They have digestive enzymes on the inside that help to recycle the materials. Most of them are going to be old macromolecules. And then it's also going to be able to go and butt up right up against that cell membrane and then expel the waste on the outside of the cell, because we don't want the waste building up on the inside of the cell. Centrioles, I mentioned before, this is only in animals. We'll talk about this next semester when we talk about replication. It is something that's going to organize those cytoskeleton fibers for splitting up the chromosomes when we get there. And this is something that we're going to focus on later, is this cell replication. The cell wall, only in plants and unicellular organisms, it is very rigid. In plants, it is made up of cellulose. In fungi, it is made up of chitin. It is usually for supporting and protecting. It gives a hard barrier, which is why when you look at a plant cell, it looks like the square boxy shape, and animal cells look very round and fluid because they do not have the cell wall. Again, mostly used for protection. The mitochondria is very important. It is in both plant and animal cells. It is what's going to take that food energy and convert it into ATP. It does have a double membrane, because it is part of the endosymbiotic theory, where we are bringing something into the cell to perform this function. It is known as the powerhouse of the cell because the ATP is made here through cellular respiration. It also has these little folds in here. If you look, this inner membrane has a whole bunch of folds. Those are called cristae. And again, anything that we have that's folding in on top of each other is going to increase our surface area. We increase our surface area, we have more room to complete cellular respiration. We also have this matrix, which is this intercellular space, and that's where the cellular respiration is going to occur. All cells have mitochondria, but we see over here that our numbers are going to increase in all muscle cells, because muscle cells are what's doing our greatest production of energy, so we need more mitochondria to help. The endoplasmic reticulum is going to be a membrane transport system. It connects organelles and helps to transport the proteins. It just looks like this whole bunch of little lines here, like a kind of a makeshift railroad or tram system. We have two types. The smooth the smooth ER is what's going to make lipids and remove toxins. The rough endoplasmic reticulum, it has ribosomes attached, and we know that the ribosomes are going to create the proteins. The Golgi apparatus is the UPS of the cell. It's going to be something that packages these proteins into vesicles. It modifies the proteins and repackages them so that they go to where they are needed. So they're going to be assembled inside the cell. This Golgi apparatus is going to move it towards the outside of the cell to go where it is needed. Cytoplasm, we said, is all the jelly-like stuff that's in the middle of the membrane. It's going to help to suspend the organelles and just give it that nice fluid environment, a little bit stronger than water. The chloroplasts are only in plants. Chloroplast is the main organelle that goes through the process of photosynthesis. Inside of a chloroplast, there is something called chlorophyll. Chlorophyll is what's going to capture that energy, and we're going to learn about this a little bit later on too, more specifically with the photosynthesis. It also has that double-bound membrane because it was part of the endosymbiotic theory where we brought this into the plant into the plant cell in order to go through this photosynthetic process. So anything that's photosynthetic is using light energy to transform it into usable energy for the plant. The vacuole. We have vacuoles in both types of cells. Smaller in animal cells, there's a large central vacuole in the plant cell. It's a storage vacuole. Basically, it keeps all of these different materials, carbohydrates, waters, sometimes wastes. We said in the plant cell, it's going to need to be filled with water, and that water is going to create something called turgor pressure. Turgor pressure is what allows this vacuole to push up against the outsides of the cell wall, and that's going to keep it in that rigid shape that allows those plants to grow up towards the sun. In the vacuoles that are in animal cell, there are usually multiple of them, and they're smaller. Again, these are something that's going to contain salts or waters or proteins and waste and things like that.